Hello everyone, my name is Ziliang. So in this poster, I'm going to present my study for identifying genomic regions and evaluating the epistasis controlling stem rot resistance in peanut. The stem rot in peanut is caused by fungus and it can cause significant economic loss. In 2015, it cost over $50 million loss in the US. So the most common way to prevent this disease is by applying fungicide, but it's not economical and it's bad for our environment. The most promising way is to breed the disease-resistant cultivars. However, the genetics for stem rot resistance is poorly understood and no genes has been cloned. Recently, a 58K SNP array is designed in peanut, which can be used to genotype the whole peanut genome and for the QTL mapping. Here in this study, we used a population with 146 recombinant inbred lines, which was derived by crossing between a resistant and a susceptible parents. And our experiments were conducted in the two major peanut growing states, Florida and Georgia. To fully characterize this disease, we used different inoculation methods and phenotyping methods. And to fully get a comprehensive analysis of the QTL, we used three softwares to do the analysis. So with this population with a large variation of the resistance, we firstly want to figure out whether those variation is caused by genotypes or not. The calculation of heritability tells us that most experiments, the heritability is over 50%, which means those variation is caused by the genotypes. With that in mind, we constructed the genetic map with over 1,000 markers. From the figure 1, we can see that the blue lines indicate the markers that are evenly distributed across the genome. So with this genetic map, we identified over 33 QTLs, and each of them can explain on average 10% of the phenotypic variation, which are presented in the figure 1 as a red line. Most importantly, 13 of them can be repeatedly detected in multiple environments, which means they're pretty stable. We also identified 12 pairs of epistatic QTLs. So the epistatic QTL means those QTLs can interfere with each other. And normally the contribution of one QTL is affected by another. Surprisingly, those QTLs can contribute to over 20% of phenotypic variation. In our QTLs, we identified over 6,000 genes, including 193 disease-related genes. Whether those genes are identified in those regions by chance or not, the enrichment analysis shows that some of the terms like chitinase or fungus disease-related genes, they're actually enriched in those regions and played a role for the resistance. Then we compared our QTLs with the previously published data, and it shows that 7 of our QTLs are overlapped with those results. So even with different genetic background, our QTL are still valid. In conclusion, the stem rot QTL disease-related candidate genes identified here can be used for the future genetic studies for the stem rot resistance. And the resistance-linked markers reported here can be used for the molecular marker assist selection in the breeding program. And the epistasis effect is analyzed here for the first time. So considering those epistatic QTLs in the future can further increase the selection accuracy. Thank you.